Welcome to this video. This is Basic Genetics Part 2. I'm Susanna Heinze with Science with Susanna, and this video will be useful for you as you are studying to prepare for the TEAS test, or if you are taking a general biology class and needing to learn about Mendelian genetics. So go ahead and watch Basic Genetics Part 1 if you're just joining us, and the rest of you, welcome to Part 2. Okay, so what we're looking at here are Gregor Mendel's experiments, and we're going to be adding some more terminology from what I did in the previous video. So let's go ahead and look at this. We've got, these are representing seeds. So we've got round seeds here. Pick the wrong pen, I guess. Round seeds. And they were crossed with wrinkled seeds, or plants that made wrinkled seeds. So when Mendel would take the sperm of one plant and put it onto a female plant, then we call that cross-pollinating. And so that's what he did, and he used a little paintbrush to do this. We're going to call the round seeds and the wrinkled seeds the phenotype of the parents. So phenotypes are the physical appearance, and these parents were, they were purebred. So we're going to call this a purebred parental generation cross. And other terms for that is that they were homozygous, that they were true breeding, and then the third term, purebred. So these are all ways of saying that the round seeds were purebred and the wrinkled seeds were. I'm going to actually write that here too, just to make sure there's no confusion. Now, when something is a purebred like that, then it's going to have two copies of the gene, like you do for everything, but they're going to be the same forms of the genes. So we're going to use letters to represent those. And in the case of the round seeds or wrinkled seeds, we're going to use the letter R. So this purebred round seeds got big R, and the purebred wrinkled seeds, its genotype is little r. So we're going to put that right here, the parent genotypes. So that's a new word in the notes so far on basic genetics. So genotypes is going to refer to the actual forms of the genes that were inherited, and we use letters to represent the different traits. So I'm going to actually start down here some key terms for this page. We'll continue adding to that because we've been doing that on the previous pages too. So we're going to say that a trait or a gene is represented with a letter. And then you can pick really whatever letter you want, but we tend to pick the letter of the dominant form of the trait, which in this case is round seeds. So the dominant form of a trait is represented with a capital letter. So, for example, we're using the letter R on this page, and capital letter R, whereas the recessive, that's the wrinkled form, the recessive form of a trait, is represented with a lower case letter. And in this case, then it's little r. And the genotype for a particular inheritance pattern is going to be the 2 letters inherited for a trait. So this is genotype is big R, and this one's little r. Let's go ahead and highlight those also in yellow. This is a new thing for this page. Now, I already had introduced a couple of terms for you. When they have the same letters, so see these plants always have big R, then we say that's homozygous. You've used that before. But we can use another word that really helps our readers know what we're talking about, and that would be the word dominant. So this organism is homozygous dominant. I'm going to highlight that in orange because I did show you that on a previous page as we've been building our knowledge. And then do you see how this one would also be homozygous? But instead, to tell it apart from this one, we say homozygous recessive. So that's what the wrinkled seeds would be like. And then some other terms that are important for you to know, but we have covered already, is homozygous, true breeding, purebred, and we actually talked about the parental generation on a previous page. That's the first generation that you are studying in a genetics experiment. Okay, so now when we look at the offspring of this cross, we call this the F1 generation. That's another term that we've covered now. And the F1 generation will 100% of it have the dominant phenotype. Because you can see that, right? All of the offspring ended up with round seeds, not a wrinkled one in sight. So you have 100% of the dominant phenotype. And not only that, but because they all got a big R from this parent, and then it was crossed with this parent, and they all got a little r, they actually all have the same genotype too. Do you see this? When they were cross-pollinated, they got big R from one parent and little r from the other parent, so they're all big R, little r. So 100% of them are hybrid. And remember, that means that they have 2 different forms for the trait, big R, little r. Or we might say that they're heterozygous. So this is F1 phenotypes are all round seeds. And then the F1 genotypes are all big R, little r. So the phenotype, all round seeds. And the genotypes, all big R, little r. So now we're going to cross 2 of these offspring, and that F1 generation cross, in this case, we're going to be calling this, whenever you have 2 hybrids that you cross, it's called a monohybrid. So we call this a monohybrid. And mono means 1. And you now know that hybrid means that they got 2 different forms of the trait for that gene. So in this case, they have big R, little r. So they're both hybrid for the trait. And the one we're talking about then is these round seeds for their phenotype. But they all have big R, little r. So then these would be cross-pollinated in order to get some offspring here. So a new term, monohybrid. And we'll try to fit that down here. Monohybrid cross. And this is when both parents are heterozygous for the trait. This should be a Y. Heterozygous for the trait. So then when these are crossed, these parents that are heterozygous, look at the offspring. Now, first of all, notice that when Mendel did this F1 generation cross of two hybrids, he didn't anymore get all round seeds. Now we start to see that occasionally you're going to get a wrinkled seed. And in fact, the only way that the offspring could have that is if it has two little R's like that. So that one's wrinkled. And then all of these offspring could be, a combination of big R, like it could have got a big R here. It could have got a big R and a little r like that. It could have got a big R and this little r. So anyway, there's different combinations, and I'm going to show you Punnett squares on the next page. But this is called the F2 generation, and this is the recessive form of the trait. The specific one we're talking about, round or wrinkled. It's only expressed and we'll say in a phenotype if the genotype is homozygous recessive. If there's any big R, then the organism in this case is going to have round seeds. So the phenotype here, this one is wrinkled, and the rest of them are round. So what's interesting is you can look at a round seed and you don't necessarily know what the genotype is. It could be big R, or big R, little r. But if you looked at a wrinkled seed for pea plants, you know that its genotype is little r. So let's go ahead and write here F2 phenotypes. So we've got these round, and wrinkled, and then the F2 genotypes. We've got big R, or big R, or little r. Those are the options on those. So we have used these terms before. This part should not be new, and the fact that we did this F1 generation cross. But there's a new ratio that we start to see then. And this could come up on the T's, or if you're taking a biology class, they might ask you this on an exam. So the idea is if you do a monohybrid cross, like this monohybrid cross, the result is always going to be 3-to-1 ratio for the phenotype. And I think you should memorize that. So 3-to-1 ratio will display the dominant phenotype, which is round seeds. But your teacher could give you any example of different traits. As long as you know which one's dominant, if you cross two hybrids, 3-to-1 of them will display the dominant phenotype when two hybrids are crossed. And that's going to be what we see with simple Mendelian genetics if you just have one form of the gene that is dominant to the other form. There are more complex ways of inheritance, but this is looking at classic Mendelian when you have, for each trait, one form of the gene is dominant and one form of the gene is recessive. So I just want to box this in. Our key terms right here. Let's go ahead and highlight that. These are our new terms for this page. So you've got a monohybrid cross, genotype. The recessive form of a trait is represented with the lowercase. The dominant is represented with a capital. And the trait itself is represented with a particular letter. Okay, so I think we'll go on to the next page of the notes. So if you like to take pictures of what I've done, you can go ahead and grab a picture of that right now. And then next we will do a page on Punnett squares. So a Punnett square is a square diagram. I have two of them shown here for you that's used to predict the probabilities of different genotypes of a cross. So let's write that here, that it's a square diagram that's used to predict the probabilities of different genotypes of a cross. So this is the definition of a Punnett square. Let's highlight this in yellow. And the reason it's called a Punnett square is because in about 1905, a man named Reginald Punnett devised these little cute squares to help predict classic Mendelian results in figuring out inheritance. So one thing that you have to know when you are thinking about setting up one of these is that an organism always inherits two copies of each trait. Each copy is called an allele, and that's what we're going to represent with letters. An allele. So the next thing to think about is what's called the law of segregation. This is one of the 3 laws that Mendel came up with. The first is law of dominance, that in simple genetics, for each trait, one form of the trait is dominant over the other. So the example I'm using, round seeds are dominant over wrinkled seeds. So the law of segregation is that when the sperm and eggs are formed, those 2 copies that you have get separated. So the alleles, which is just a technical term for the forms of a trait, and I'm using the words trait and gene interchangeably here for simple genetics. These alleles will separate during meiosis. So the mom's egg will have one of her copies, and the sperm will have one of the copies that came from the dad. So the gametes only get one of the copies that their parents had. They don't get both. So each gamete, and that gamete is a word to indicate either eggs or sperm. So we can look here and see that if this is what the purebred mom had, if she had big R, all of her eggs are going to actually end up with a big R. And if this father here, being purebred, he is going to cause all of his sperm to have little r. So I just chose, to have the father be the one with the wrinkled seeds. So this is the sperm from the male. And then this represents the egg options from the female. And this is an example. This particular parent generation cross is 2 purebreds. So we're going to use pink for the eggs. So let's say that, and then blue for the sperm. So this sperm right here could fertilize this egg, and then we would represent that by putting them in the same box together. So see, we put this big R in here with this little r, like that, see? So then this would be the plant that grows. This is an option for the offspring. So here are the parents, and then this is the option for this particular offspring that will grow up, and it will produce round seeds. You can see that here. So then if this sperm fertilize this egg, then we would have another big R and another little r. So do you see it actually is the same? And then if this sperm were to fertilize this egg, then we would have big R, little r. We always put the capital letter first if there's going to be a capital letter, just to make it look tidier and more easy to read. And then if this sperm were to fertilize this egg, then we would have one big R, little r. So this Punnett square is a visual way for you to see and understand why 100% of the offspring that we already looked at on the previous page will have display the dominant phenotype. And that will be the F1 generation, will display the dominant phenotype, which is round seeds for this trait. And not only that, but 100% of the offspring will be heterozygous. So see how their parents were purebred, but once you cross them, then they're not purebred anymore. Kind of like I have a dog who, her name's Tulip. Her dad is a golden retriever and her mom is a cocker spaniel. So they were both purebreds, but now she is a hybrid between a cocker spaniel and a golden retriever. So these seeds are hybrid. So 100% are heterozygous. But you wouldn't know that just by looking at the seeds, because this is going to make round seeds and the parent made round seeds. So, you know, you can't tell whether it's big R, or big R, little r just by looking at the seeds. Now, if it's wrinkled, you can tell it must be little r, because we know that wrinkled is recessive. So let's go like that. Okay, so these are terms that we've already used. Talked about dominant phenotype. We've talked about heterozygous. Now let's see what happens with the F1 generation cross. And what we're going to do is called a monohybrid cross. So monohybrid means one trait. We're just looking at round or wrinkled. And hybrid means that they're all, so this would be one trait, seed shape. And they're all big R, little r. So this is what the cross will be. And these would be the sperm options then. And these are the eggs. We'll put pink for the female, blue for the male. And then we'll go ahead and fill these in again. So if this sperm fertilizes this egg, then once again, we're going to have big R would be what this little plant's genotype is. Now this plant, it's going to have a capital R and a lowercase r. So it has a different genotype, but notice they would both still have round seeds, right? But this one is a hybrid and this one would be a purebred. And then here, if we have, remember we always put the capital letter first. So there's another hybrid. And then look at this last one though. We get little r. And this explains why 25% of the offspring would display the recessive phenotype. So the results of this monohybrid cross is that 75% of the offspring are going to display the dominant phenotype, and that will be round seeds. And it's a 3-to-1 ratio, so remember that. And the recessive form will show up in 25% of the offspring. So the recessive phenotype was masked when the purebreds were crossed, but it reappeared when the hybrid offspring were crossed. So that's going to be the end of this particular page. And I'm going to do a basic genetics part 3 and cover what are called monohybrid and dihybrid crosses. So I'm going to be going through dihybrid crosses in the next video. But And we'll cover the last law, what's called law of independent assortment for classical Mendelian genetics. If you want to go to my website, I guess I can put this on here. So sciencewithsusanna.com. I have all my free resources there, and I have the handout so you can color along with me. I have a lot of Quizlets and practice questions there as well. All right, I will see you in the next video.